I just told you that I got involved with establishing a relation between genes and proteins long time ago under the mentorship of Professor Tatum. And our job was to, he had a collection of mutant neurospora. Neurospora is a fungus, and our job was like to find a needle in the haystack in the way that to find which protein is defective in that particular mutant and how it influences its phenotype. At that time at Rockefeller, there were many groups working on proteins, and they had different approach, and I'm glad to tell you that almost all of them got Nobel Prize for their work. Anyway, I'm going to give you a brief history of the science of omics. In reality, when we started the work, the science of omics, or even the word, was not coined. And only much later, Gunter Blobel, who is himself a Nobel Prize winner, professor at Rockefeller, told me that Nawin, what we were doing at that time was the infancy of proteomics. So anyway, I will proceed with that. And I will just summarize you three things that led to the advancement or coming of the science of omics. And these three things, I'm sure all of you know that the most important thing was the Mendel's law of inheritance, which established that how the genes controls a phenotype. And the second advancement was by Beadle and Tatum, who shows that how the genes or DNA controls the protein, and kind of integrated chemistry to biology. And finally, the whole thing was crystallized by the work of the structure of DNA by Watson and Crick. And then later on, and one of the Nobelists at Rockefeller, Christian de Duve, told me that Nawin, everything else was just a matter of detail, and you can imagine what a great detail it was, which brought Nobel Prize to several dozens of scientists in molecular biology and genetics. So, and of course, these details were the advancement of different technology, like recombinant DNA technology, and the recombinant DNA technology, protein DNA sequencing, PCR, and thoroughfoot technology, gene editing, CRISPR, and bioinformatics. Well, I might mention that science of omics was defined by Lederberg. Lederberg was a student of Tatum and shared Nobel Prize later with him. And I'm not going to the rationales why Lederberg thought that omics is originated from a Sanskrit word om, which encompasses the complete description of a system or universe. Well, I will proceed to the different, you can see the different omics, you can describe them in genomics, epigenomics, proteomics, or branch of that, interactomics. I will dwell on that for a moment, and metabolomics. Now, I will just tell you very briefly about genomics. Genomics has been, I will not go into detail about genomics because genomics is a very vast field, but it did recognize in many organisms, I will refer only in terms of the human, that it established the number of the genes, the copy number variation, and single nucleotide polymorphism. And it also provides the basis for the understanding of what makes us human. there are essentially 5 to 6 genes which I will come, I will show you in a moment. And of course, the coming of the synthetic genetics. And here are the few genes. There are at least 5 genes, like the one which led to the development of brain, then when the FOX protein, which or gene which caused us the ability to speak, then the amylase and many other proteins that distinguishes us from any other primates. And the proteomics started with one gene, one enzyme concept and mutant or disease organism and defective or missing proteins. And the clearing example was hemoglobin that time. And you can see there are mutations, and you can see the mutant hemoglobin is here with sickle cell. And then with the work of Finson and others of denaturation and renaturation, they established the 4 level of organization of the proteins. And then, with the help of Tatum's own work as well as Garrod and others, they did establish the defect in the metabolic pathways leading to different diseases, like phenylketonuria, which all of you are aware of that. And then it also establishes the collinearity of gene and proteins, that the sequence of DNA corresponds to the sequence of amino acid in the protein. And then the sequence of protein was first attempted by Edman, who was also at Rockefeller. He was a Swedish guy who was doing postdoc with Stein and Moore, and there he started this. In this, he can label the very first amino acid with a chemical isothiocyanate and cleave it and then recognize it. And in this way, a number of amino acid in the protein could be recognized. And I must tell you that the proteomics flourished with the development of throughput knowledge technologies such as 2D gel electrophoresis, developed by O'Farrell in Berkeley, and by Klose in Germany at the same time in 1975. This may surprise you that O'Farrell work was rejected to be published by the Nature, but that all sometimes has happened in the history of science that people have. Here is the 2D gel of people suffering from Alzheimer's and a normal human. And then finally, the sequence of protein or amino acid protein was established by a number of physicists who developed the mass spectrometry. And in this, they could establish which amino acids are there by their weight, and they were determined by the time of flight. And in this way, you can see that there are 8 of them and how they were established. And the length of the fragment can be written as this amino acids. Now, the proteomics also provide the basis for understanding the differentiation. You know, long time ago in Germany, people have established that during the development of insect, there are changes in hormones which does bring the thing, but they were not sure what was actually happening. But with the proteomics, you can see this is the caterpillar, and this is the silk moth, and you can see that the conversion of one to another is due to, even though they have identical genomes, it is due to the differential activity of the proteins, which is projected here. The red ones are the one from silk moth, and the blue ones, or it may appear black to you, is from the caterpillar. So it was a great finding. And then you can see one of the great anomaly in science of genetics has been that why we have in different size of genomes, but our complexity is not correlated with the size of the genome. And this was done by interactomics, or interaction between protein and proteins. And this can be done in vitro as well as in vivo. This is mostly the work of Stumpf from Berkeley, in which he shows that you can see rice has almost the same size of genome, about 2.5 billion nucleotide. Human has about 3 billion nucleotide, or in terms of gene, it has 3,300. This has 2,300. I forgot to mention that. But you can see the interactions, interactomes, the amount of protein-to-protein interaction in a human is more than a log more than in rice. So, and as you can see that as the lower the complexity, the less and less is the interaction among the proteins. So this has been a very important finding of proteomics to show that it is the interaction of proteins, it is not the size of the genome that determines the complexity of organism. Now, from this, they have come up with the idea of diseasomes, in which you have different human disease network and different genes involved, and then you can go to their complexity. And then, not yet finally, the proteins are very important as biomarker in human diseases. One of the reasons is the protein itself can act as a drug, or it can be a target of a drug. So it is very important for designing the drugs for the treatment of diseases. And here is a list of proteins which can be used as a biomarker. And in this, one of the important things has been that there is not, at times, you cannot rely on only one protein as a biomarker, but sometimes you can take a number of proteins as a biomarker of a disease. Yes, you can see here. And then the proteomics has brought the possibility of personalized medicine. Personalized medicine means that for every individual suffering from a disease, a particular regime of treatment can be developed for that patient. Not when you know the current approach in medicine has been what is called one size fits all. This has been going on for quite a while. And one of the important examples is the cancer treatment in which you go through surgery or radiation and chemotherapy. But because of the better understanding of cancer, that has changed and it has developed into a very targeted use of drugs. Now, you can see that like you see, there are specific drugs for the cancer, tamoxifen, glycovan, Herceptin, and but we know we can with the Proteomics we can devise better. Like we know the cancer patients metabolize tamoxifen in a different way. Tamoxifen is broken down to endotoxin with cancer fighting ability, and so on and so forth. In the same way, the gefinin and many other of them have the same way. Herceptin is effective only in patients with HER1 mutation. If people don't have this mutation, the Herceptin will not work. The same is true about the cardiovascular diseases. Like the dosage of warfarin can be adjusted with a personal genomic background. In the same way, in myocyte, in obesity, there are two kinds of myocyte has been identified, and one is the brown, which burns fat, and the other, which is the brown cells, which is a zinc finger protein, and it stores the fat. So by targeting either of the two, you can perhaps control the obesity. But I tell you, there is one guy, I'm forgetting his name, at Rockefeller, who devised the leptin or discovered the leptin. He once told me that, you can control the weight or obesity in mice, but you cannot control in human, because human has no sense of stopping when not to eat. And we all know that human is the only organism which can override its own biochemistry. So it not always necessarily follow the rules of biochemistry. So now I will mention you about metabolomics and other omics. Metabolomics is a very good approach to understand diseases and treatment, because in case of human, you can see there are 23,000 genes, there are about 100,000 proteins, but there are only about 18,000 metabolites. And so by figuring out, because metabolites number is very little, you can figure out that how you can control a disease. And in common parlance, all the time in all cultures, it is known that we are what we eat. And that's what I'm talking about, the metabolites. Now, finally, the epigenomics, or environmental modification of DNA, and as well as protein at times. And this has been done by the work of people in Israel, as well as in this country, in Drosophila, in mice, as well as now in human, that if people have been deprived in some way or other, their DNA can be modified. Most of the modification is because of methylation. And after in certain circumstances, these altered DNA can be passed on to future generation, and that can cause the problems. So the epigenomics is a very important field, and lots of attention is being paid to that. In epigenomics, you can also think about the genomes of other organisms that are within us. And like many bacteria, some of them are very friendly bacteria and some of them that are not friendly bacteria. And also don't forget about the genes that we can harbor or we can get and harbor from our pets. So there are many things. I'm not against pets or anything, but I just want to point out your attention to that it can cause epigenomics. So anyway, in somewhat conclusion of proteomics, advances in science of omics, particularly protein, will lead to understanding of several complex diseases, a great prospect for personalized medicine. I can give you a couple of examples. One is the one which is developed in, I think, in San Diego, where they have extracted a factor, BDPN, and injected into people with neurological disease and cured it. So that is a big advancement, and I'm sure similar things will come. So you can see these guys, Trudinsky and others in the University of California, San Diego, I was telling you, they have discovered things. Well, this is not moving, so that is causing the BDNF, a neutral factor, which can, injection of that can do that. In the same way at Wellcome Trust, Sanger Institute, they have found a large number of proteins, about 130, controlling about 130 brain diseases. So I have presented you a kind of summary of the omics, or particularly proteomics, from its infancy to maturity. And I'm sure, as the talk previous to me and many other talks to come, you will see the sophistication. Now, this is a painting by Picasso, and I'm very fond of Picasso because Picasso always has depicted or expressed the agony of mankind. And this is, you can see a girl in front of the mirror, and she is very much dismayed about her future. And I reflected on that in 1975 when we were having a conference in Essilomar to pause on to consider the pros and cons of the recombinant DNA technology, because recombinant DNA technology, it became certain about the changes that might be introduced in human as well. But I must tell you, it has been about 40 years, and we have learned how to use science with very judiciously, and we have not yet caused any disaster, and we have no reason to be. But although I must say that the Picasso's concern about human agony due to the social and political upheaval is still on this earth. And I don't think I can dwell on that anymore. Well, in the end, I must thank Professor Tatum for giving me this opportunity to bring to this science of proteomics, and many agencies which supported my research. And finally, I will I must thank my wife who had always supported all endeavors in my scientific efforts. And not only that, right now I have some medical condition that I cannot drive for a long distance, and she offered to drive more than 400 miles to bring me yesterday. And so I'm very much thankful to her. And with all this, I can end my talk. If you have any question, you might as well raise it now or later. I'm going to be here 3 days anyway. We'll be having lots of gathering, lunch, and many other things.
So now I'm inviting Dr. Kulesh to give the memento to Dr. Nawin Mishra.
Well, thank you so much.
Our pleasure.